Hey there, pre-meds. Welcome to this video where we're going to be covering a tricky genetics topic, siRNAs and microRNAs on the MCAT. I get asked about these guys a lot, so I want to make a video about what they are and how they're most likely to be tested. For your note-taking purposes, you'll put siRNAs and microRNAs in the biochem subcategory, biotechnology, tools, and research. 1B for those that are following along with the AAMC categories. You'll often get tested on these in the context of research design and data interpretation, so skill 3 and 4 questions. Don't be surprised if this comes up once in a while in Chem Phys, because this is a research tool that we often use for a variety of different studies. Okay, so first off, what are microRNAs and siRNAs, and what's the difference between them? siRNAs and microRNAs are both small sequences of RNA, so think nucleotides, and they have uracils instead of thymine bases. And they're about 21 to 25 nucleotides long, so super, super small. Functionally, we can think of them as honestly the same thing when we're using them in research studies. They have the same general function. However, if you would like to know the difference between them, microRNAs are usually endogenous, which means they're naturally occurring in our body, where siRNAs are often exogenously produced, manufactured in a lab, and placed into cells for research purposes. There are some more nuanced differences there, but for the purposes of the MCAT, we can think of them as functionally the same. So what do these things do? Well, they are small pieces of RNA that are complementary to specific messenger RNA, mRNA sequences, and they hybridize or kind of complementary bind to the sequences of mRNA out in the cytoplasm. To understand the impact of that action, we'll need to go back to our central dogma of genetics. You may remember that our central dogma is all about how we make proteins for our cells, starting with our DNA blueprint. And what we do is we start with DNA, which is our blueprint in all of our cells, and in eukaryotes, that's taking place in the nucleus, which I've drawn here. When we want to make a protein, the first step we do is known as transcription, right? So transcription is where we make our mRNA sequence from our DNA. So we use RNA polymerase to transcribe our DNA into RNA. Before it becomes mRNA, it goes through what's called post-transcriptional processing. I'm going to just write that as PTP here. All right, post-transcriptional processing is only in eukaryotes and includes things like splicing out the introns, adding a 5' methyl cap, and adding on a 3' poly A tail. All of this is to say we go through this pre-RNA to mRNA sequence in the nucleus, and then it's messenger RNA, it's mRNA ready to be translated out in the cytoplasm or in the rough ER, depending on if those proteins are staying in a cell or leaving a cell. And it's going to go through translation with proteins known as ribosomes. We have ribosomes that read our mRNA sequence and use that to translate it into a primary amino acid sequence to make our proteins. It's important to note it's not relevant for this particular subject area, but we do usually go through a post-translational process after the initial translation to make our final proteins. All right, so we usually do post-transcriptional and post-translational processes. Okay, so now let's think about how hybridizing a microRNA or an siRNA with our messenger RNA can affect this central dogma. So you may remember that our messenger RNA is single-stranded, which means it's just one strand of nucleotides. I'm going to make our nucleotides just these little dashes here. So this is our messenger RNA, and it needs to be single-stranded because the ribosome reads that strand and brings in tRNAs that are complementary to 3 nucleotides to choose the right amino acid. But what happens if we have a microRNA or a siRNA, which I'm going to write in red, is that hybridization happens before we get to the ribosome, right? Remember, they were complementary sequences. So now this microRNA and siRNA have now made the messenger RNA double-stranded. And if it's double-stranded, it cannot be translated. All right, so we're not going to get a protein from this mRNA. In fact, it usually gets degraded by cell systems once it becomes double-stranded. So what is our function of our microRNAs? I'm just going to write miRNAs and siRNAs. Well, they they're inhibitory, inhibit translation. All right, and I'll draw that right here. They inhibit here. Now, the important piece, and this is why we're kind of working backwards through the central dogma, even though I'm sure this is kind of basic for many of you, the important thing here is that it's not affecting, the microRNAs and siRNAs are not affecting transcription or post-transcriptional processing. Anything before the translation step is still happening and is happening as normal. So we're getting the same amount of messenger RNA, it's not mutated, we're doing splicing still, right? We're still doing that transcription step with our RNA polymerase. None of that's being affected until we get to the step of translation, and that's where the inhibition occurs. And knowing that this is the part of the process that's being inhibited is often the piece that's tested on the MCAT. So we're now going to do 2 MCAT style practice questions on these topics to really reinforce the application of siRNAs and microRNAs. Before we do, I'd like you to go ahead and subscribe to this channel. My goal with this channel is to provide you with videos on all of my most commonly asked MCAT questions to help you along your prep. I also do have a few resources that are linked below. One is an MCAT study hours calculator, and one is an MCAT skills assessment to see if you need to work on things like math on the MCAT, research design, and data interpretation. So please feel free to check out those resources and subscribe to this channel. And now we're going to go ahead and do a practice question. Okay, so this is a question that you may encounter on the MCAT about siRNAs. So go ahead, pause this video, give this question your best chance, and then we'll go ahead and review it together. See you in a minute. All right, let's navigate this practice question together. First, it says the research team wants to use siRNAs in their experiment testing of a certain protein. They plan to compare protein levels with and without this siRNA. Which of the following experimental procedures would be appropriate to make sure the siRNA was functioning as intended? Okay, so a couple pieces here. The first piece is that we're just trying to see if our siRNA is working. So what would indicate if our siRNA is working? We would have decreased translation, right? That would mean that it's working. So we need to have some sort of system of measuring whether or not we're decreasing translation, aka decreasing proteins, right? Because our product of translation is protein. So if our siRNA is working, we should have less proteins. So really this question is asking which of these 4 procedures will allow us to measure protein levels. Now, we didn't talk about the different blots in this video. That will be another video, but we'll go ahead and review each of them together right now. So a northern blot is used for RNA. So it'll allow us to visualize different RNA sequences of different sizes. Southern blots do the same thing, but for DNA. So it allows us to visualize DNA strands of different sizes. And Western blot allows us to visualize proteins. All right, so maybe we're already like, okay, we know the answer. PCRs are systems that allow us to amplify DNA. All right, so PCR is also related to DNA. It allows us to amplify certain DNA sequences. So we have an RNA visualization, a DNA visualization, proteins, and then another DNA amplification sequence. Now, I know it's tempting to go for northern blot because you're like, well, this is about siRNAs. This would measure siRNAs. But a northern blot would only show us that we have siRNAs, not that they're working, right? It would just show that they were there, but it wouldn't necessarily show us if they were working. How do we know if they're working? We have to look at the outcome, and the outcome is going to be whether or not we've decreased protein levels. So what we're looking for is a way to measure protein levels with and without siRNAs, because that's going to let us know if it actually decreased translation. And the only one that matches that is our Western blot. C. All right, so check your own skills for this question. Did we need to review the different blots, but we understood we were looking for a protein measurement? Or did we get tripped up by remembering that northern blot was RNA and it got us kind of tripped up because we were talking about siRNAs and not thinking about looking for the outcome of the system instead of just what we have? So go ahead, review your notes, feel free to pause this video, and now we're going to move on to our next practice question. All right, so for this practice question, you may need to make your screen full screen to see all of the graphs and figures. And this is one that would be passage-based. So I'm bringing in some passage-based information alongside answer choices that are graphs. So this is a more difficult question type that, again, you're likely to see on the MCAT about siRNAs and microRNAs. So go ahead, pause this video. I would give yourself 2 to 3 minutes to do this question, and then come back and we'll review it together. Okay, so let's review step by step with the information that's provided and then how to answer this question. It says the majority of dietary vitamin A is stored in the liver and then is transported to the organs as retinol by its carrier protein, retinol binding protein, or RBP. This is very typical of the MCAT for them to give you some background information before introducing us to the testable information later in the passage. They're now telling us that the retinol forms a complex with RBP known as holo-RBP. By the way, holo as a prefix for enzymes and proteins usually means active form of. So holo-RBP is actively doing its job of carrying the retinol. So that holo-RBP now goes to STRA6, which is a membrane receptor for it, and it allows us to take in retinol up in the cells. So we can take a note here, so it may be good to practice some note taking here. So we have paragraph 1, they're telling us, okay, we have holo-RBP, right, which allows us to take in retinol, and that is going to attach to STRA6, which is receptor, and that's going to get us into cells. All right, that's going to get us into cells. Great, so far so good. And it uses this fancy transport mechanism, right? They're saying, okay, it's like this unique thing, and it has a high affinity, and only one retinol is allowed to bind at a time, and the RBP stays out of the cell. So it's not the RBP that goes in, it's the retinol that goes into the cells, right? And then they said endocytosis inhibitors do not prevent this uptake. That's usually a hint if the inhibitor doesn't have an effect. What that means is this is probably not endocytosis. So the receptor is using some other process that's not endocytosis. So far so good. We have some background information. Now we have the question. Researchers measured the retinol uptake in untransfected and STRA6 transfected cells, and we looked at the presence or absence of retinol bound to either RBP or bovine serum albumin. So there's a couple of pieces here that I want to mention. The first is that untransfected just means we haven't messed with it, right? So it's just going to be like a blank cell. It does not have, so no STRA6, right? Untransfected, no STRA6 versus yes, STRA6, right? So we've got those as one set of independent variables. This is what we're manipulating. The second set of independent variables is whether or not we have retinol bound to RBP, which is its transporter, or if it's bound to bovine serum albumin. Now, bovine serum albumin, just for your information, is a typical protein that we see in blood, and it often is a carrier protein, but it's not going to do any specific action. So what they're using this here most likely for is for a control, right? A negative control where we're not expecting any kind of binding or uptake to occur for this BSA. That's usually the case unless we're actively studying bovine serum albumin. All right, so we have whether or not we have the receptor as an independent variable, whether or not we have the binding protein as an independent variable, and our last independent variable is the point of this video, which is whether or not we have an siRNA that is specifically targeting STRA6. So remember, STRA6 is a protein, and we have to think about what do we know siRNAs do. siRNAs, as we remember, decrease translation of proteins, right? So that means we should get less protein. So if we have siRNA, that means less of the STRA6 receptor. All right, so if we have siRNAs, we have less of the STRA6. If we don't have siRNAs, we should have normal levels, right? So that should be our normal control. All right, so now let's put this together to answer this question. I know this can feel overwhelming to try to figure out all these variables in graphs as answer choices. So my trick is to just go one variable at a time, one decision at a time, and just eliminate what I know to be not true. So let's start with whether or not we have the receptor. Yes, no, STRA6. So if we do not have the receptor, should we get uptake into the cell? If we don't have a receptor, can we get retinol into the cell? Probably not, right? So we can say, okay, for the untransfected cells, the ones that do not have the receptor, I should see low levels of retinol uptake. And our retinol uptake is on our y-axis. So I'm looking for the black bar here. I'm looking to see low levels across the board because if we don't have the receptor, we can't get that retinol into the cells. So A looks good. We have those low levels for the black bar. B looks good. Low levels for the black bar. Ooh, C. C says it doesn't matter if we have the receptor, which doesn't make much sense because how else would we get our retinol into the cells? That's out of scope, right? This would say that the receptor doesn't matter, right? Which we know it does because they told us in the passage it does. So let's go ahead and eliminate C. And now we're left with D, which also has low levels for untransfected. So relatively straightforward decision. We weren't expecting to get all the way to the right answer with that one. but we've at least eliminated one answer. So now let's go to our second independent variable, which is whether or not we have the binding protein. And if we remember, our STRA6 is the membrane receptor not for the retinol, but for the binding protein, RBP. So if we don't have RBP, we shouldn't get binding, which means we shouldn't get much uptake. So for BSA, my second prediction is the BSA graph should be low, right? My BSA graph should also be low, because at least lower than RBP, because we don't have that binding. So A, it's like, it's still there, but it's at least lower than the RBP, so we'll keep it. B, oh, that's a problem. See how this says that it doesn't matter if we have RBP or not? We do know it matters. In order to bind to the receptor, we need our RBP, so we can eliminate B. And D looks good. D says that BSA, when we have BSA, we have a lot less uptake activity, right? So we're now down to A and D, right? A and D both look good. So now let's move on to our third independent variable, our siRNAs. So remember, when we have siRNAs, which is plus here, right? Plus means presence. So when we have siRNAs, we should have less of the receptor. So it kind of matches up with our initial independent variable of no STRA6, right? So what did we predict for that? We should have less uptake. So I'm saying, okay, when I have siRNAs, I should have lower uptake than if I don't. So the problem with A is A says it doesn't matter if we have the siRNA or not, which doesn't make much sense because we know the siRNA should be reducing the amount of the receptor. So we can eliminate A, and then D matches beautifully with our prediction where we have a lot less uptake when we have the siRNA, and it honestly matches nicer with the BSA having very, very low levels. So D is our answer here because we were able to work through each variable and find the only answer that makes sense. All right, if that was a challenging question for you, that is totally fine because it was meant to be. And I hope you took away some key lessons from our practice. Number one, utilizing content with a couple clear rules. siRNAs inhibit translation, can help simplify a really complex research setup. Secondly, when you're moving through answer choices that have graphs, try to go just one variable at a time and eliminate rather than trying to jump to finding the right answer. It's really easy to convince ourselves that graphs tell us something that isn't necessarily true, so it's much better to do an elimination strategy for those types of questions. I hope this was helpful for you. Please feel free to check out our next videos, and as always, happy studying.